Okay. Assalamu alaikum. Welcome everyone to another episode. Today we are going to discuss about finished product analysis in QC lab. So in this video we are going to discuss about the overview of the tests that are being done in any sort of QC lab. Beyond all these tests, there are also many sort of other tests that are very much product specific, but these are the common tests. Now let's start with description. In case of description, what is it? It is that description of that product, of that finished product. For example, a tablet may have scoring in which we break down the tablet and eat in pieces, or it may have different colors or coating materials, or the packaging, or that color and clarity of any sort of injections. All these are description part. This is very important part because this is being watched or noticed by the consumer. Consumer can see if the description does really matches with the product actually. If there is any sort of clarity issue or color issue, consumer can easily identify that, and that is not very good for any sort of pharmaceutical industry. Okay. In the second place, we must ensure that identification is okay. So in case of identification, what is this thing? This is, we all know that any sort of finished product is made with API. What is API? API means active pharmaceutical ingredients. This thing actually does exert that finished product therapeutic effect. Okay, the therapeutic effect is being exerted by this API. Okay, so we must ensure that the API is correct. Alright, in case of pantoprazole tablet, we don't have any sort of paracetamol in it. Okay. So to do identification, generally we use IR, UV, HPLC in sort of chemical analysis, or sometimes AS is used for identification purpose if there is any sort of metal ion. These are widely used process. In case of IR and UV, we use spectrums to identify. We make spectrum of that raw material as a standard, and we take spectrum of that finished product, and then we compare with these two spectrum. If these two spectrum matches, then we can say that this finished product contains that same raw material. In case of HPLC, as we all know, HPLC have peaks, chromatograms that have peaks. If the same standard have peak in, like for example 5 minutes, and the sample also gives spectrum in 5 minutes, then we can say that this finished product contains that same raw material. This way identification is done. Let's move to assay, the next very most important aspect of raw material finished product analysis. So what is assay? Assay is the average content of that finished product. Average content of that raw material or that API in that finished product. For example, when we say this paracetamol tablet is 500 milligram paracetamol tablet, this implies that this tablet may have a weight of 500 milligram or 600 milligram, but in this 500 or 600 milligram tablet, there is 500 milligram of paracetamol in it. Okay, we must ensure it in QC lab through different type of chemical analysis. Mostly we use HPLC, UV, and different type of titration. Okay, sometimes potentiometer is also used, and sometimes AS is also used if different type of metal ions are present. Next, we move on to physicochemical characteristics. Let me give you some example. For example, we test pH. We all know that in case of injectables, this is directly injected into our system, okay, in the blood flow. So our blood flow have a very specific pH. If any other different pH is inserted in the blood flow, it might be lethal also. So pH is very important for us to maintain in that finished product. Similarly, osmolality, we have a very specific osmolality for our blood, and our product must match of that osmolality. That's why we have to test. There are also specific gravity, sedimentation rate if we use a suspension. So to denominate the suspension quality, we must see that sedimentation rate. Also, there is water content. Water content is very important for those products, for the solid products actually. During manufacturing of tablets, we go through wet granulation, then we add some water. So if the water content is very much, then there is a high chance of microbial growth in that preparation. So we must control that water limit. Generally for tablets it's below 4% or 3%. and so on. In case of lyophilized product, water content is much more important, as because lyophilized product, if it has much more water content, then what will happen? It might degrade. So we must actually control the water content in lyophilized finished products. Let's move on to impurity. So impurity is any sort of admixture or impurity in that finished product. It can be of two types, the related substances and impurities. Now, related substances, as I have discussed earlier in raw material video, can be of the origin. So imagine this is a paracetamol API. So this paracetamol does not exist in the nature. We have to manufacture it in the synthetic way. In the synthetic manufacturing system, there is a precursor chemical that is being converted into something else. Then after reaction, it converts into something else, and then finally it may become paracetamol. Now, all those raw materials that are used to manufacture this final product, these are all related substances. And in the final product, some of those trace amounts of those chemicals may be present. These are impurity as related substances. Also, this paracetamol can degrade. It can degrade to become some other chemical during our shelf life period. Okay, so we must actually control the related substances quantity in that finished product. In order to do so, we use generally chromatographic technique like HPLC, GC or TLC. Why chromatographic technique? Because related substances are of very trace amount, and if we cannot separate it from that raw material itself, we cannot actually determine the quantity of these related substances. Another class is impurities. So impurities are these may come from manufacturing process. For example, in the same compression machine, we have manufactured pantoprazole, and after that we are manufacturing esomeprazole tablets. After changeover, some trace of pantoprazole may come into esomeprazole tablet, or from the dust in the corridor or in the room. These are all impurities, or it may come from any other sources. So we must contain these related substances quantity and impurity quantity within a very strict specification limit. Okay, next move on to preservative content. This is the specific test for specific products. Generally, liquid product contain preservative, some sort of preservative, to prevent microbial growth in that product. Okay, as we all know, preservative is not good for human body, so we must contain or must control those preservative content up to a certain limit. It's kind of we can say kind of assay, and generally we use HPLC to determine the preservative content. Okay, so next move on to content uniformity. What is content uniformity? But before understanding content uniformity, we must understand assay. Okay, so in case of assay, what happens? I'm giving you example of tablet. In case of assay, we take like 20 tablets, crush all the tablets all together, make a blend, and from that blend we sample a certain amount of powder. And after that powder we take weight and make sample and analysis using different sort of analytical tools. The thing is that in case of assay, we are getting average result, average of 20 tablets, okay, or 10 tablets, whatever the sample size is. It doesn't necessarily mean that all tablets have same amount of that API. For example, if I say I have 500 mg of paracetamol, that doesn't mean that in the strip 10 individual tablet contains 500 mg of paracetamol. It may vary. Okay, to ensure that each tablet contains the similar amount of that API, we must perform content uniformity. In case of content uniformity, what we take is we analyze each tablet individually. Generally, the sample size is 10. That means 10 individual unit is being tested. After test, we can show that all the tablets contain almost similar amount of API. We express it in a specific statistical tool. I'll discuss it later in some other videos. in case to test this content uniformity, we use UV, HPLC, potentiometer, sometimes say yes, all these sort of tools. These are actually same as assay. The tools are same as assay, but in case of content uniformity, we test individual units. Now, according to USP, what we do, if the tablet size, not tablet size, the API is more than 25 milligram in each individual doses unit, or the percentage of API is more than 25% of that finished product size, then we can perform weight variation to ensure content. Okay, so for example, metformin hydrochloride, this tablet contains 500 mg of raw material in each tablet. In that case, we can actually weigh all these tablets, like weigh 10 individual tablets, and ensure that 10 individual tablets weights the same. As the API is much more in these tablets, we can say as the weights are same, that API are also similarly distributed in all these tablets. Okay, this is content uniformity or weight variation. Expression is the same as content uniformity, that statistical expression. Okay, we express it in acceptance value or AV value, which must have to be less than 15. Okay, let's move to final parameter, that is product performance. Okay, to ensure product performance, we perform certain tests. So what is actually product performance? Let me show you an example with dissolution. Okay, imagine that we are taking a paracetamol tablet. We're actually swallowing it, so it must disintegrate into our body or into our stomach. This is disintegration. If it doesn't disintegrate, then the API inside those tablets won't release in the system, and it won't be dissolved in the gut fluid, and it can't be extracted in the bloodstream. Okay, so disintegration is our first parameter, that is that product performance. Okay, how well that product actually does disintegrates. Second one is dissolution. Okay, tablet disintegrates, but does it dissolves in the gut fluid? To check this thing, we perform dissolution. In this case, we use dissolution tester. Generally, this tester is a jacketed vessel, ensuring 37 degrees centigrade temperature. This is the temperature of normal human body, and there are certain vessels in which there are some sort of fluid, some sort of buffer or so. There are a paddle or basket or any other arrangement. There are 7 arrangements in pharmacopoeia, so we can use some sort of arrangement like this and place that tablet or unit doses form in that vessel and test. Okay, so the test is the tablet must have to disintegrate and dissolve in that fluid. It's much like dissolving sugar in water. Okay, if the sugar is dissolved in water, it will disappear. Like that, the tablet will disappear in the solution, and we'll test those solution to check if the raw material has been dissolved or the API is dissolved in the system. Okay, there is also NGI or ACI. These sort of impactors are used to check inhaler quality. Okay, so if we take certain like DPI or powder inhalers, so are these being absorbed uniformly in the body, or this particle size is okay? We must check these through NGI, ACI or any other arrangements. And to check all these things, there is also perfusion, like for example, if we test patches that is being used in the skin, so it must perfuse and actually perfuse through the skin. At the rate we have to ensure that it meets the acceptance criteria. This is also another product performance. One more very important thing about dissolution, that in case of dissolution, we actually simulate the product bioavailability in vitro or in lab conditions. Okay. Also, for example, there are products are enteric coated, like tablets like pantoprazole tablets are enteric coated. So we intend pantoprazole tablets to disintegrate in the intestine, not in the stomach. As we all know, when we take tablets, it first goes to stomach. Their pH is the pH of stomach is really low, and in this environment, if the tablet is disintegrated, then the pantoprazole will be destroyed. So in order to keep the tablet intact, we use enteric coating. So here in the stomach, during the stay in the stomach, it doesn't disintegrate. It will disintegrate once it releases into the intestine where the pH is much higher, and here the tablet is released or disintegrated, and it systematically absorbs. So during dissolution test we can test all these things. So these are the basic tests of finished product analysis in a QC lab. Apart from this, there are also microbiological tests. In microbiological test we test pathogen and viable bacterial count. Different type of, it depends on that product content, or sometimes we use particle count if there is injection or so. So hope this clears the concept about finished product analysis in a QC lab. Wish you very good luck, and bye bye.